Hello, everyone, and welcome to this Anabios webinar. My name is Chris Mathis. I'm the Chief Commercial Officer at Anabios. And as people enter the virtual conference room, I do have a few opening remarks, and I'll start by saying, Raf, thanks so much for joining us. Really appreciate you taking the time for this webinar.
Thank you, Chris, for inviting me, and it's great to be here.
Also in the virtual studio is my colleague, Gary Watkins, who's the Director of Marketing. And I'd really like to thank Gary for putting this webinar together. In fact, Gary is behind the scenes on all the webinars, and he talked me into doing these starting at the end of 2019, before the pandemic. And that really set the stage for us to be able to provide these virtual conferences sessions throughout the pandemic. So thanks for that, Gary. One of the things that you've done also in the past year is set up the Anabios YouTube channel. Can you tell the audience about that?
Sure. Yeah, we have about 60 videos currently on our YouTube channel. We have a number of different speakers, including Rob Schwartz and Dr. Ted Price. Most of these videos are our webinars, as well as videos from our translational research conference that we sponsored last year. You can visit our channel by going to youtube.com/@sign, the @ and Anabios Corp.
Thanks a lot, Gary. So we have quite a few people that have registered for the webinar today who are probably fans of Raf. Thanks to us tagging him on the LinkedIn, we've got a lot of people here that may not know about Anabios. So before I introduce Raf, I do want to spend a few minutes talking about our company. And we have been in the human tissue and primary cell business for over 10 years, but really in the past couple of years, hepatocytes. And I want to explain what makes us uniquely suited for the hepatocyte field. And so what I can tell you is that Anabios is a CRO, and quite a unique CRO too. We are headquartered in San Diego, California. We also have a site on the East Coast in Philadelphia, and a site in Washington State, not too far from Seattle, that is the location of our subsidiary Cell Systems. Anabios recovers human organs from ethically consented donors, and we use the tissue or cells to conduct physiological assays in which we can test preclinical compounds for drug discovery and safety pharmacology projects. Anabios also leads the industry in providing biologically relevant human tissue samples and primary cells for translational research. We have essentially redefined first in human, and working with Anabios is as close as you can get to a clinical trial without actually doing a clinical trial. Another thing I want to mention before I introduce Raf is that we do have in the GoToWebinar panel towards the bottom, there's a place where you can write in questions at any time during the webinar. There are also a couple of brochures on Anabios, hepatocytes, and LSEX from Cell Systems that you can download in the handout section. So now it's my pleasure to introduce Raf Witek, who's a highly accomplished interdisciplinary scientist and people manager with over 24 years of work history in industry and academia. He's a hepatologist and expert in cellular liver and GI biology with skills that cover cell and molecular biology, pharmacology, ADME, tox, biotechnology, stem cell biology, and advanced 2D and 3D cell culture, including 3D printing. Currently, Dr. Witek is a freelance consultant supporting liver biology and biotechnology for preclinical development and the advancement of therapeutic cell and gene therapy technologies for liver failure and liver metabolic disorders. Prior to becoming a consultant, Dr. Witek was the VP of Translational Sciences and the head of the liver program at Aspect Biosystems in Vancouver, BC. During that time, he led the development of 3D bioprinted implantable liver microtissues for treatment of liver failure and monogenic metabolic liver disorders. Prior to Aspect, he was a director of cell therapy platform at Ambus Medicines in South San Francisco. In that role, he was developing CMC manufacturing processes for hepatocyte-based cell therapy. His contributions range from the procurement of human liver tissue to the optimization of hepatocyte isolation and purification workflows. Dr. Witek, his extensive career also includes a significant period at Thermo Fisher Scientific, where he served as senior staff scientist, senior manager, associate director of R&D, ADME, and toxicology. He led hepatic R&D teams, optimized hepatocyte isolation processes, and developed hepatic media and reagents using design of experiment methodologies. He also made substantial contributions to the development of cryopreserved primary, NPC, and iPSC differentiation to hepatocytes. Dr. Witek earned his PhD in interdisciplinary medical sciences from the University of Florida in 2005. And in 2007, he was awarded a senior postdoctoral research associate in our NRSA F32 fellowship in gastroenterology at Duke University Health System. In addition to his scientific training, in 2013, Dr. Witek completed an MBA in strategy and business development at Duke University. So Raf, we're really excited about this presentation today, entitled Considerations in Testing Using Primary Hepatocytes in 2D and 3D Cell Culture. Take it away, Raf.
Awesome. Well, thank you so much, Chris, for the introduction. And also thank you, Anabios, for having me here today for this webinar. So for today, what I decided to do is split the webinar into 3 sections. The first section, we're going to cover introduction to liver cellular biology and considerations for selection of 2D and 3D cell culture. So this is relatively straightforward and basic information. However, a lot of times people they have a difficulty trying to figure out what they should do or use, whether they should work with 2D or 3D. So hopefully this information will provide you with enough knowledge so you'll be able to choose which applications you wanted to work with. Next, we're going to talk about hepatocyte variabilities and why is testing necessary prior to using primary hepatocytes. You know, in my field, primary hepatocytes are very, very variable, and it's known that you always test every lot before you purchase. However, a lot of times when I join different groups or when I consult with different groups, that is very new knowledge for them. So I wanted to really talk about this too, so everyone can understand what are the variabilities, where they're coming from, and what you can do to prevent them or at least minimize them. And then finally, I'll talk about primary hepatocyte testing strategy and how you can find the best cells for your research and how you can optimize 3D aggregation in case you were doing that. So with that, I will start with just the basic biology of the liver. So liver is the largest solid organ in a human body. It has a lot of liver functions. It has a lot of functions in general. So then one of the main ones is synthesis of serum proteins like albumin, alpha-1 antitrypsin, others. It also do bile acid synthesis. So bile acids, for those that don't know, those are essentially byproduct of metabolism that liver excretes and removes from the body, and those usually go to the intestines and helps with the digestion process. Next, they're also doing detoxification of xenobiotics, of which the byproduct is obviously the bile. They're also doing intermediary metabolism. You have amino acids production, lipids, and carbohydrates in the liver. Because of the size of the liver itself, liver is also storage for glycogen, for lipids, for vitamin A, iron, other things as well. And also because of the large size and large volume of blood that goes to the liver, it is the primary organ of phagocytosis. So the cells that are mainly responsible for that, you have 2 types of cells in the liver. You have a liver parenchymal cells, which essentially means the main liver of the cells. Those are the hepatocytes. So those cells, this is exactly what we're going to talk today about. Those are the ones that do most of those functions that I just listed. But they also have other cells. Those are the liver non-parenchymal cells, which essentially means everything else but the main ones. So that includes Kupffer cells, the liver macrophages. So they usually involved in a phagocytosis, in the removal of debris. They're involved in liver regeneration, involved in the inflammation, for example. You also have hepatic stellate cells. Those are usually accessory fibroblasts that help maintain hepatocytes. They're producing ECM in the liver. During activation, they produce collagen and lead to the fibrosis, and they're part of the NASH or MASH in a liver injury. You also have the liver sinusoidal endothelial cells, LSECs. Those are the cells that line the sinusoids, and hepatocytes are stuck right between them. They're the ones that control what hepatocytes see, how much microvilli sticks out into the bloodstream to pick up the nutrients. There are also the cells that can sometimes close the fenestration that they have and then prevent access to the hepatocytes. So they're essentially controlling the metabolism. They're also, for many that don't know, they also involve in a partial metabolism. Some of the metabolites actually go to sinusoidal cells and they go back to hepatocytes. So a lot of times having some L6 impurities in your hepatocytes can actually help with the metabolism of toxicity. And then finally, you have a biliary cells, also called cholangiocytes. Those are the cells that are involved in the removal of the bile from the liver after the metabolism is completed. So now going to the next level of the liver, if you go to the histology, the single unit of a functional unit of the liver is called hepatic globule. So each hepatic globule is composed of one central vein surrounded by 6 portal triads. And between portal triads and between the central vein, you have a sheets or layers, or sometimes people call cords of hepatocytes. And when you can see on this picture, in the middle, and I'm actually showing you that the hepatocytes are not actually equal. There are zone 1, zone 2, and zone 3. So what actually happens in the liver is when the blood comes to the liver through the portal triad, that blood comes directly from the heart, directly from the lungs. It's full of oxygen. So a lot of oxygen reaches to the first immediate cells that's surrounding this portal triad. And those are usually referred as zone 1 hepatocytes. They're involved in the oxidative metabolism because of this oxygen level. And they're also very small. They're usually single nucleated. They're really nicely packed. They're typically small cells. Then if you go on the extreme of that, toward the central vein, you have those large, gigantic hepatocytes. They're usually called zone 3 hepatocytes, or just the metabolic hepatocytes. At that point, there's less oxygen, and most of the metabolism that happens in zone 3 is essentially the xenobiotic metabolism as much as glycolysis and lipogenesis. Now, what's interesting thing about those hepatocytes is that in zone 3, they're large. In zone 1, they're small. And why is this important? Well, the reason why this is important is that if you're doing the isolation of hepatocytes, when you work with human liver, with adult human liver, that organ is very large. And if you're perfusing that organ, if you're perfusing this liver, you try to inflate it so to the point where you get enough enzymes that gets to every single cells within that liver itself. Well, the problem is that because the organ is so large, a lot of times you only get to zone 3 and maybe some of zone 2. Rarely the adult livers would generate zone 1 hepatocytes because of that. And the reason why this also works like that is because of the large hepatocytes. The area that they represent on the outside is fairly large for digestion, so they are separating first. By the time you get to zone 1, your zone 3 will be dead. Since most of the isolations is set up to generate high quality cells, you stop before you get zone 1 hepatocytes. So if you're in a business or if you're working in an area of research where you are interested in zone 1 hepatocytes, you need to be aware of that. The majority of adult hepatocytes that you get from the vendors will not contain many of the zone 1. There are mainly zone 2 and zone 3 because they are most metabolically active. If you're interested in getting zone 1, you need to look at pediatric livers or younger livers. Because of the size, they are smaller, they are easier to perfuse. As such, you also get the more even distribution of cells with them. So it's important to know, that's why I wanted to mention this. So just be aware of that. That majority of the cells you're gonna get is zone 2 and zone 3. If you're thinking about longevity studies and long-term studies, you need to look for the different source of cells, and that's pediatric or smaller livers. So now moving to each individual unit of the cell, well, essentially hepatocytes. So when you look at the image of the hepatocyte that I have on the left, you can see there's a lot of stuff happening inside the cell. Number one, you have 2 nuclei. So hepatocytes tend to be polyploid. They have multiple copies of chromosomes, and a lot of times you can actually see that they may be 2 and 4 and 8 and 16, and they will have usually either large nuclei, which will be called a megalocyte, if it has this kind of a nuclei, or it just have 2 nuclei, and that's just usually predominant. The look of the cells that they have those 2 nuclei. The reason why they do that is because DNA serves as a template to make the RNA, to make the protein. And hepatocytes are essentially large factories that produce a lot of stuff. And if you have a large lines of production with a lot of RNA, DNA, RNA, and then protein, that makes the cells to have a large output. Because of that, you see that you have a nucleus, you have endoplasmatic reticulum, you have a lot of Golgi complexes inside the cells, you have a lot of mitochondria. All these processes, they need energy. So hepatocytes tend to be busy. When you look at them under the microscope, a lot of times you see those granules on the inside. Those granules, this is all the stuff that they have. So on the right side, I have this picture of the zone 3 hepatocyte. Those hepatocytes are typically called heavy hepatocytes because they have so much mitochondria inside. They essentially by the density, if you like put them on the density gradient, they're gonna pellet at the bottom If you wanted to separate them, there are ways to separate them because of that weight. So they are really busy cells. And because they also they're so busy and they have so much stuff inside and doing so much work, they also develop this very intricate way of how they exist within the liver itself. So typical cells, they will have one basal surface on which they attach to the matrix, and then the apical domain will be on top of the cell. With hepatocyte, it doesn't work exactly like that. So in the middle, you have here an image where it shows you have 2 layers of endothelial cells, the L-sex. Right between them, you have an ECM, which is essentially the space of this. And then hepatocytes are attached to that space of this, to all the collagen and fibronectin and other proteins that you have in that space with their basal surfaces. And you can see there are two of them, the one on top and one on the bottom. And the apical surface is actually forming on the inside, almost like channels, like piping. And that piping contains the bile, that is the excretion from those hepatocytes. So why you have this kind of structure is because with so much work that those cells do, they need to have a lot of uptake. And being able to provide them uptake on top and the bottom of their basal surface allows to get a lot of nutrients inside the cells, and then the channels on the inside can push those cells out. So when we culture those cells, typically you're trying to reproduce this kind of environment where you have an ECM on both sides of the hepatocytes. So on the left here, you have an example of the basic 2D sandwich culture of hepatocytes, which is the current state of art for culturing those cells. It's a very simple system. It's a plate with a collagen type 1, typically red type collagen that you have, on which you plate your hepatocytes. You plate your hepatocytes at 100% monolayer. So you have to have the confluent monolayer on them. At that point, you will overlay them with the gel tracks, and you end up with essentially a sandwich of hepatocytes stuck between two layers of ECM. And over time, they will form into bulk canaliculi, and they will have all these apical surfaces develop, which is called polarization. I'll talk about this in a second. But if you wanted to do something cooler, and you really wanted to get the uptake going from both sides, one way to do this is to actually plate hepatocytes on the transwell sandwich culture. And in this example, you take the transwell with the membrane that has pores of about 0.2 or 0.4 microns. You coat that with collagen type 1, and then you plate hepatocytes on top and you overlay them. And then if you submerge that in the bottom plate and you put media on top and the bottom, and then you put your substrate on both sides, you end up with this kind of a polarized, or not polarized, but the bidirectional uptake, which is great for those cells, and they will work just slightly better. They're not much better than regular 2D, but at least they represent another form of the culture that you can try. Well, this really plays cool role is that when you think about liver, in the liver you have, with hepatocytes, you have a separation between blood and bile. So the blood is carrying nutrients to hepatocytes, and the bile takes the byproduct out. And the bile is, a lot of times you refer to bile as bile salts. So the cool thing about the transwell that you can actually create that type of a membrane polarized excretion. So if you assume that your blood has less salts and your bile has more salts, you can create media that has more salts on top. That's where you put your substrate. And then you have a bile media that contains more salts on the bottom. And then what you will end up having is that during the metabolism, your byproduct of metabolism, your metabolite, will actually end up in your bile part of the dish, which is a really cool way to mimic this if you wanted to do it. Outside of that is just a more complex system. If you wanted to learn more about those systems, we actually published a paper back in a couple years ago. Actually, let me see, where did I think on my, yeah, there it is. So here's the paper about Ed Lacluse, myself, Mel Anderson, and Mark Powers, organotypic liver culture models meeting current challenges in toxicity testing. It is older paper, it's from 2012, but it's a very nice paper. You will see a lot of those figures that I have in this presentation in this paper, and it will teach you about a lot of this kind of model systems if you're interested in them. But going back to our culture, so this 2D plated system is great, but there's a better way of doing this, a better way of generating better functional hepatocytes, and this is 3D. So one way to do this is to create a spheroids, and spheroids is essentially nothing else but a ball of cells. You take few cells, you bring them in a close proximity to each other. If you give them time to aggregate, They will form the aggregate within about 5 to 7 days, and then you give them a little time to mature. They become very functional cells. So the systems today that you can use to aggregate hepatocytes like this are, there are the variety of this kind of systems. The basic ones are like the Corning plates or Thermo Fisher plates. Those are ULA or the low attachment plates. They're U-bottom or V-bottom. They're a great system. You simply put the cells inside, give them a few days, they aggregate, they form a spheroid, and you can use them for your studies, for your research. Another approach is from Inspiro. They have this beautiful plate, Acura. The really cool thing about this system is that, number one, the changing media in a thermo or Corning plate, it can be very challenging if you don't have some kind of a instrument or biomag or some kind of a liquid handling instrument for this. So if you do it by hand, the Acura plate is amazing because it has the little latch on the side of the well that you can simply bring the tip to that latch, and then it allows you to change media without disturbing the spheroid. The other thing is, if you try to image cells on a U-bottom plate or V-bottom plate, it's a pain. It's very difficult because the bottom is not even. So getting a nice image of the spheroid is very, very challenging. Well, Inspiro came up with a really cool idea. They created almost the optical flat bottom, and when the spheroid lands on it, there's nothing that distorts the image, so you can get a very nice images using their plates. Another way to make more spheroids is to go to stem cell technologies, and they have a plate called AgriWell. It's a microwell plates. Instead of making one spheroid per plate or per well, now you can make hundreds per well. So another approach, they have different sizes. So if you wanted to make spheroids of 25 cells, 50 cells, 100 cells, they have different applications for that. And then finally, if you're looking for a manufacturing of spheroids in large quantity with very easy user friendliness, so PBS Biotech have this vertical wheel bioreactors. So the cool part about this is because you have a movement of liquid in two kind of directions, what happens is you can control the size of your spheroids with simply adjusting the speeds of how the media is mixed and then playing with the density of your cells. The other cool thing is that you can stop the system for a few minutes, which allows the cells, all the spheroids to settle, and then you can easily media change in them if you wanted to do so. One of the biggest challenge of keeping those cultures for a really long time is this media changing. So PBS is a great idea or great way to do this kind of stuff. So you have different ways to do that, to do this kind of a 3D, but why would you want to do that? Well, the reason why is the function itself. So here's just an example of what could you get by looking at all those different systems. So the gray shaded area that represents just regular plated hepatocytes without overlay. So this is like the worst of the worst in this case. And then the white one is the 2D Geltrex overlay. So this is the sandwich culture I showed you. Blue one is the transwell, and then 3D is in the red. And then assume that all the data that you see here has been normalized for the cell number. So obviously you have to do that because in the spheroids, you have a much smaller number of cells that you're evaluating versus when you have a plated cells. So on the top data set, this is the gene expression. You can actually see that when it comes to albumin secretion or albumin production, there's not much really difference between plated, whether it's a transfer or not transfer. But then when you look at the spheroids, you see nearly double increase expression of the albumin. Similarly for the phase 1 enzyme, in this case CYP1A2, you can see there's not much difference between the transfer or Geltrex, but there's a huge increase in the 3D, and the same thing on phase 2. Now, this I'm only showing you representative here for phase 1 and phase 2. We actually evaluated almost all major CYPs for phase 1 metabolism, and you can see in many cases you see exactly the same thing. And then obviously this is gene expression data, so you would want it to look at the protein. So on the bottom you have actual LCMS analysis. We're using phenacetin conversion to APAP for CYP1A2, and then in the bottom you have hydroxylation of testosterone to 6-beta hydroxytestosterone. And in this case, you can see exactly the same thing. There is some improvement in a transwell versus just a regular Geltrex overlay, although it's not much of a huge improvement. The biggest improvement you see in the spheroids. And notice one thing, that all this activity really went up on day 5. And that is the reason why when I showed you originally how you make those spheroids, you want to wait until this day 5, and then you want to give them a few more days so they can normalize. So what happens at that point, and I wish I had this figure made differently in the past, but basically what happens is after day 5, this activity goes up and then it normalizes, and the cells become very consistent when you want to use them. On the other hand, if you wanted to use the spheroid at day 3 versus day 5, you see how much of a difference you would have in your data if you do analyze like that. So there's the reason why you'd want it to mature your spheroids. Now, before we go to really like how you choose this and how you do it, one thing I want to talk about is talk a little about the function and what you can actually expect from the systems. And a lot of times people don't realize this, but when you think about the activity in the human liver, it is actually really, really, really high. And that activity in vitro is almost impossible to really reproduce. And that is the reason why you have all those 3D cultures, why you have the microphysiological systems, why you have those dynamic flow bioreactors, because each one is trying to improve and get it closer and closer to this whole liver function. So when you take the liver and you isolate hepatocytes from them, and then you don't do anything else, you just simply use them as a suspension, you end up with suspension hepatocytes, and you can see the activity drops, and it drops really, really low. But they're still useful. They still have the residual activity from the original liver. They have enzymes, they have proteins, everything is working just like if this was in the liver, except they don't have the supportive other cell types. They don't have the media or the blood flow with all the oxygen tension and whatnot. So because of that, the function is significantly decreased. Still, you can use it for metabolite ID, MET-ID, you can use it for clearance, you can use it for transport. Albeit those are very short cultures, you can use them only for maybe 2 to 4 hours before they die. Because of that, you want to play them. Well, when you play them, the function goes even lower. And the reason why is because you play them in a media, in an artificial environment. You put them in some media that contains some salts and amino acids, and it's close, it works, but it's not reproducing exactly what you'll see in a human liver. Despite of that, you still get about 5 days in this type of cultures. You can still use them for metabolism, clearance, but now you can also use them for induction and transport, because this requires, or some of those studies require more time. The mechanism of action or toxicity can be done better on 2D plated hepatocytes. And then when you go to the 3D, you increase this by simply increasing the length of culture. Now you can go for 10 to 30 days. Now you can do those studies even maybe better or similarly. And also you add another ways of generating data for disease modeling. Before you couldn't do this type of research. Just think about this. If you're trying to build a fibrosis and you're putting hepatocytes with stellate cells, can you do this in 2 days? It's going to be very, very challenging. But if you do this in 3D, now you can keep the culture for 14 days, for 21 days, or maybe even a month. And during that time, you can actually see the expression of or production of ECM and the position of the ECM, and they can treat with different compounds of interest to see if you can reduce the fibrosis or increase the fibrosis. Same thing for Kupffer cells. You can combine Kupffer cells with hepatocytes and create inflammation or create MASH model. With this kind of systems, purely because you get extra 10, 30 days of culture that you couldn't do before. Now, also implantable tissues. You can generate spheroids that you can now put in a 3D printing, scaffolds, or encapsulate them in a alginate or other material, and you can use it for implantation. Something that you cannot really do with plated cultures or the single cells because they simply will die fairly rapidly. So why would you want to really deal with this? I mean, this is like, it sounds like pain. I mean, the cultures don't really work well. There's so many different things and intricacies of this. And so why would you want to work with this? Well, the reason why I have to work with this is because of FDA and EMEA. Well, in reality, all potential small molecule drugs must be screened through primary hepatocyte cultures for drug-drug interactions. And that can be only done with primary hepatocytes. I think the other thing is animal testing. So when you think about doing work in animals, it's expensive. It takes time. It's a lot of training. So because of that, there's push for 3 R's: reduction, replacement, refinement. How can you replace some of those studies with in vitro? So then you can minimize amount of work that you do on in vivo. So because of that, you can generate, in vitro cultures today, and there's a push to develop those 3D spheroids, develop microphysiological systems that can kind of almost reproduce some of those studies that you can do in animals. Now, keep in mind that animal studies might be better for what your research is, but you can also develop some data from in vitro cultures faster as the kind of a directional data, how you can set up your experiment in vivo better, or how you develop your dose, or how you test your print or cell quality, for example, in vitro before you go to in vivo. So you can do a lot of work in vitro before you go to in vivo, and that is the reason why this is worthwhile. And then finally, animal models are not very predictive of idiosyncratic toxicity. And that is usually idiosyncratic toxicity because of other cell types like stellate cells that are activated, or macrophages that are activated and producing TNF alpha or other cytokines. Again, modeling something like that in a 3D system or in a microphysiological system is so much easier than trying to do this in an animal. So because of that, we do that. Now, so you want to now work with the hepatic culture. Which one should you choose? Well, it really depends on your application, how much money you have, and what you're interested to obtain at the end. So obviously your basic here is the 2D hepatic sandwich culture. This is established model of primary hepatocytes. It's been in the field for 30 years, over 30 years. Everybody knows how to do it. There's a ton of data out there. You know how your data compares to other data out there. So it's essentially industry standard in pharmaceutical drug discovery. Testing, if you generate data in the system, you can show it to FDA. FDA knows what you're showing them. If you can talk to your friends and everybody knows what the data means. It's easy to set it up. You simply get the plate with collagen type 1, you put the cells on it, overlay them, and they're ready to go. You can do a lot of high throughput screening. But you are limited by this 5-7 days to get this full metabolism. So if you can do that in the short time, that's your best system. But on the other hand, if you're looking for something that is better, then you may want to go to the 3D. Now, one more thing I'll mention here is, I don't know how many of you know, but hepatocyte culture is essentially senescent culture. What that means is that your cells are dying and you're simply trying to keep them alive as long as you can. So hepatocytes do not proliferate. When you get them from the liver, they're already in this stage where you damage them and they're slowly undergoing this kind of a slow senescence in a way. So because of that, everything you do is trying to keep them as long as you can. I think the limitation on this is the typical hepatocytes. I think if I remember correctly, in the past, I was told that live somewhere around 160 days on average. Some of them will live longer, some of them will live shorter, depending on how much exposure to toxins they have. So if you assume that it's 160, your typical culture should live about half-life of that. So you're not going to be able to really go beyond that 80 days. I'm assuming, at least, if you're doing this primary culture, because eventually they're all going to die. Well, still, going to 3D gives you this longevity, and it allows you to go maybe 20 days or 30 days, which you couldn't do with the regular plated system. Now, what 3D gives you? It offers you physiological function because the cells simply self-assemble. You don't have to really force them on the ECM and plate them on a layer. They just simply kind of get together and then make their own ECM, and then before you think, they start really function and work the way the liver would work, or you'd expect the liver to work. Now the metabolism is higher, and it allows you to do a lot of different things. It allows you to, for example, print with them, encapsulate them, and so on. Example on top, you have, you don't have to just make one spheroid. You can make a 96 plate full of spheroids. You can make 384 plates full of spheroids. Depending how you want to do it, there are systems out there that allow you to do it. Now, here are some limitations. Well, you can imagine your regular microscope may not work for your 3D culture. You need to have a confocal microscope. You need to have a high throughput system or high content screening system that allows you to handle cells that are in 3D. The cost of plates is fairly expensive. The cost of media or the media may have to be adjusted to maintain it. Because the cells are more functional, they're also using more nutrients. As such, the media will have to be optimized better, or media changing system has to be more frequent. So altogether, that represents another set of challenges, even for analysis. Analyzing small spheroids can be challenging, but it's worth it. If you know how to build them, if you know how to make them, they can be very fun to make, and they work really well. So altogether, you can, depending on your applications, you can choose either one of those. But then the one more question remains is, so why we have to do primary hepatocytes? If there's such a pain and all this, can we maybe just use the cell line? Why not to use there's a HepG2, there's hepatocytes. A lot of, in my consulting days now, sometimes I meet with the groups and they tell me they work with primary hepatocytes, and they tell me they work with HepG2s. And you're like, well, HepG2 is not a primary hepatocyte. And the reason why is because it doesn't have hepatic genes or hepatic expression of the CYPs or P450 and so on. So the reason why you want to work with primary hepatocytes is because they have a full functional metabolome. So they have intact P450 metabolism that includes phase 1 enzymes, your phase 2 enzymes, the glucuronidation, and then you have a phase 3 or transport. So you have uptake and excretion. Second, and which is very important, you have the nuclear receptors. You have the CAR, PXR, AHR that are intact in those cells. Next, you have a functional and robust ammonia detoxification, which you may not have in those other cell types. And then finally, the integrins, E-cadherin, and GAP junctions that help the cells to polarize and maintain the structure may not exist the same way in the cell lines, which proliferate rapidly and cannot really become that as cuboidal. As such, some of this mechanistic work that they do, the production of synthetic production and enzyme production and metabolism, cannot actually happen. And indeed, those cells are not exactly producing many hepatic, they don't have hepatic receptors, they don't really have a P450 metabolome. On the side here, you have some data, just HepG2 against primary hepatocyte blood. You can see there's albumin, obviously it's a hepatocyte, so it will have some kind of albumin presence, but CYP3A4, non-existent. CYP3A7, which is the immature form of CYP3A4, you would like, well, it's a proliferating cell, maybe you should have some immature enzyme. No, it doesn't have it, it's very little. UGTs, phase 2, not existent. AFP is crazy. Obviously, there are proliferating cells. You would expect them to have a huge amount of AFP. And then also there are hepatocytes, there are hepatoma line, so there are HNF4 alpha. So that is the reason why using primary hepatocytes is critical, and cell lines cannot really work for that. All right, so with that concludes my introduction to this basic liver biology. So now I'm going to change the gear slightly and just talk very quickly about variabilities and why you need to test your hepatocytes prior to using them. And the reason why is to reduce variability in your data and to increase data reproducibility in your lab. So very quickly, so there's 3 sources of variabilities in primary hepatocytes. The first one, donor variability, is well known. Everybody knows that. It's well established that genetic and environmental factors lead to substantial individual differences between people and different their metabolism and different transport. P450, polymorphism, SNPs, no mutations, all of those exist in those cells. You have to be aware of that. So if you're looking at this kind of stuff, genotyping is required to evaluate each lot, so you can control what you're actually working with and what you have. And also you have the age variability, sex variability. A lot of times hepatic function, basic screening of hepatocytes can tell you how good those cells are in metabolizing things and how variable they are from at least that standpoint. Typically, this data is included on C of A, and this is your way of how you can analyze it and at least remove some of this donor variability. So this is good, but there are 2 more variabilities that a lot of times they either disappear in your thinking or we simply don't pay too much attention to. So the first one is the interlaboratory variability. So this is essentially what you do in your lab and how you handle your hepatocytes. And I don't want to spend too much time on this one, but the reality is that we have our favorite methods, we have our different trainings, and we have ways of doing stuff the way that we like. And sometimes that creates a lot of problems because the methods are inconsistent. So a way to really improve on this or reduce this variability is, if you're in a company, standardize your cell handling protocols, develop and standardize your testing SOPs. Make sure that people follow them. Make sure that your groups are performing them the same way. And to do that, make sure that your reagents and media is also standardized. That is not some media that somebody likes it and not the one that you actually have to use it. For me, one of my biggest P in a field is when you're working with primary hepatocytes and your Williams media runs out. And then you go to the fridge and there's DMEM. And you go like, well, it's a red, it's media. Why don't you just use the DMEM instead of Williams? I mean, it's just media, right? Well, DMEM doesn't have a reduced glutathione. It doesn't have a proline in amounts that the hepatocytes need. The amount of glucose may be way too high. So the end result is you're actually creating a huge variability if you do so. So having a SOP, a good ways, and standardize them and making sure you follow the SOPs verbatim and don't change medias too much and standardize that, that's really a way to really improve your hepatocyte cultures. Next one is training plan for the new employees. When your new employees joining your team during onboarding, don't assume that they know how to work with hepatocytes. Show them the method. They might be doing something better than you, but if you're doing it in your way and your lab is consistent on doing that, make sure that everybody can do that the same way. It is fairly important. And then finally, work closely with your vendors. Your vendors are your friend. They have application scientists, they have a field application scientists that can come over to your lab and can help you with the training, help you with your cultures, help you with your SOPs. So anything that you need, to help you standardize the process and improve it and make it better, work with them. So this essentially would cover your variability. But there's one more, and this was kind of interesting one. So this one is the cell manufacturing variability. And how it works, well, the process of isolation of hepatocytes is very complex. There's many steps, and many of those steps are different from vendor to vendor. For instance, typical isolation of hepatocytes is done as ex vivo two-step perfusion, where your first step is you're using some kind of chelating agent, like in this case, EGTA buffer. That buffer removes calcium from the system, removes also calcium from the junctions between the cells, separating the cells from each other. It also flushes the liver out of blood, so there's no blood that will interfere with your second step, which is the collagenase digestion. Well, guess what? There's many different flavors of collagenase. Not every collagenase is created equal. There are purified collagenases, there are crude collagenases, there's type 1, type 2, type 4, type 8. So there's many different ways of how you can isolate the cells. Well, guess what? They are not the same collagenases. They're not the same enzymes. So each company has their own unique buffer or unique buffers, unique collagenases, and that creates a very unique process for each group. Also, whole organ versus resection. In a whole organ, you can get more cells and you can get the very good quality cells. However, it is very difficult to inflate that liver, and getting the cells in the zone 1 can be very, very challenging and also difficult to handle. At the same time, resections or cutting or processing the liver in a smaller section can be easier, but it's still the same challenge because the resections have their own challenges and their own issues, and the older livers have fibrosis and other ECMs inside the parenchyma itself, which also prevents from the full inflation and digestion. So you still end up with zone 2 and zone 3. So it really doesn't matter which one you do, although the number of cells and some of the quality could potentially change depending how to do it. There's also single pass where you perfuse it directly with just enzyme versus the recirculation. So then on the liver tissue, you have different acceptance criteria. You have different age for different tissues that you get. In our field, you can also have a tissue quality. Some might be bloody, less bloody. They could be coming from donors that are more healthy versus less healthy. Drug users, the people that drink a lot of alcohol, for example. So that is part of the tissue quality and acceptance criteria for the companies. And then the WAT and CIT, the warm ischemia time versus cold ischemia time, all of those play a role in this process. Now, I'm not going to go too much into the rest of this stuff. One thing that I wanted to mention, or two things that I wanted to mention, is it also gets to cryopreservation. So on a cryopreservation, to my knowledge, every vendor uses different cryo solution for preserving their hepatocytes. They all have their own secret sauce that works for them well. And a lot of times it contains more or less the same components. Typically, you'll have with some kind of permeating reagent, in which case almost always it's a DMSO. And then you have a cryoprotectant, which can be either PEG-based, starch-based, dextran-40-based, or some other way. But some of them will work better in some applications. Some of the other ones may not. Some of them may coat the cells little. So, for example, when you're doing transfection or gene delivery, you may inhibit the getting the genes inside the cells versus the other ones don't. The other one is handling and storage of hepatocytes, and this is extremely, extremely important. You have to keep the cells in the vapor phase, liquid nitrogen, not in a liquid, not in a dry ice, not in a -80 freezer. And the same thing for shipping. You cannot be shipping hepatocytes on liquid nitrogen or dry ice. If you do so, you end up killing about 20 to 30% of your hepatocytes. Whether you're storing them or you're shipping them, that killing will remove that percentage of cells from your prep and potentially damage remaining cells, potentially impacting the quality of the isolation. So altogether, you see there's a lot of different steps that those vendors do differently from each other. This is not exactly standardized process, and it's very different. But that is actually advantage to you as a user, because that creates what I call a unique cells for each vendor. And because you as an end user, you have so many different applications, so many different ways of how you want to play with those cells and use them. Testing cells from different vendors, you may actually discover that someone might be doing something better that actually makes, or something that makes your cells better for your application. And in my case, I had those instances where I would get cells from one vendor for one application and they'll perform beautifully. And then next time I would change my application to something else, and those cells from that vendor would just not work at all. And sometimes this could be due to the processing. Sometimes this could be simply because the process changed in their shop. Because of that, industry developed very precise analytical methods to help standardize this kind of selection for the end user. So you get C of A for every lot of hepatocytes that's generated by the vendor. And they'll tell you a lot of information about this lot that helps you choose it. The second thing that they also do is it is fairly accepted in a field that you can request a free or with reduced cost test vial if you're purchasing large quantity of hepatocytes for your in-house testing. And the reason, and you should do that, you should really test the cells before you actually use them for your research. So with that, I'm going to move to the final section, which is how we test them and what you can get out of them. And then in here, I actually included the pictures from the next slide. Those are actually hepatocytes from Anabios, and I just love this lot. Every time I look at this picture, I just cannot get enough of it. So on top, you have a plated primary hepatocytes from that lot at like 5 days, and you can see there's just beautiful monolayer with almost no dead cells. And on the bottom, you have this beautiful bulk caniculi staining with cDFDA. Just shows you the quality, and just love it. So I just figured I'm going to include it. But the reason I wanted to go here and talk more about this is, so the first way of how you can evaluate your hepatocytes is to look at the C of A for each company. So because this webinar is done for Anabios, I went straight to their website and I pulled one of those lots, exactly the lot that I love, and to show you an example of how a C of A looks, how you can read it, and how you can use it to your advantage. So C of A will have multiple informations on it. You don't have to go crazy about this. You just have to look at a few things. So first thing, viability. You don't want dead cells, so you want to look at the good quality viable cells. So in this case, 90%. 90%, that's great. If the counting is within 5% error, that tells me that's somewhere between 95% to 85%. That's really good quality hepatocytes. So viability is great. Second, cell number per vial. Well, you don't want to have 10 cells. You want to have enough cells that you can actually use them. A typical plate requires 10 million hepatocytes to really plate. So if you have 10 million cells per vial, that's perfect for your one plate applications. If you're working with 3D, that might be equivalent of 30, 40 plates of 3D hepatocytes. So you don't need as many, but having more cells is always good. So in this case, 10 million, I love it. 10 million is my magic number. I love that. So the next, going to the donor demographics, you have some information. You need to know whether that's a pediatric or not pediatric, whether it's a male, female, if there's any kind of aberrant things, or like how the patient died or drug use. You wanted to see that. So this is included here, so we know that this is pediatric lot. It could be part of the reason why it's so nice. But then the other thing is the representative images, and this is, the pictures are worth 1,000 words. Those pictures will tell you exactly how those cells look. Now keep in mind that just like you as a researcher, you pick the best quality pictures for your presentation. It's the same thing on the C of A. Those are best quality pictures. So you have to judge them with the grain of salt. So if they're perfect, that's great. But if they're not so perfect, they're probably the best of the best they could find. So that's how it can tell you right away whether you're looking at the good quality lot or not. In this example, you have a hepatocytes at 5 days. They look amazing. They actually show you at 14 days that they can last even longer than typically, 5 to 7 days. This is 14 days. This is phenomenal, great. cDFDA staining shows you this is perfect transporter lot with such a nice expression of bulk caniculi. And even better for me and for my research is 3D forming. So this is great. Now, in addition, they also provide you with enzyme induction activity, metabolic activity, and there was a third page that I did not include here. So this is also useful if you're looking, for example, for specific, metabolism at a specific level or specific induction. So this is great. So that means that you're ready to buy it, right? Well, not exactly. So just because this looks so great and the C of A looks amazing, it doesn't mean that those cells will perform the same way in your laboratory. So because of that, I like to use this term, trust but verify. You need to verify that what they generated in their space with their highly trained people, with their standardized methods, you can do the same thing in your own lab. So the basic testing is request the testing vial, and then when it arrives, first thing you do, you test cell number and viability. Do they match the C of A? They should match the C of A within 5% error, maybe 10% error. But if they are way off, then there's your first red flag. You don't want that. Second one, you plate the cells on collagen type 1 with a plating density of about 0.8 to about 1 million cells per ml. Standardize that. You always want to have the same number. I typically use 0.8. I know some people like to use 1 million because it's easier to count. But typically just standardize that. So when you plate them on the plate at a 0 hour, so you can actually put the cells on the plate, you give them a little good shake, put them on the microscope, let the cells settle for about a minute or two, and you can look at them without disturbing them too much. You will see actually cells settling. And so what you're expecting to see is you want to see almost like a single layer of cells with enough space around them, because number one, there is another 20 to 30% cells that are still settling. You don't see them because they're floating, and you don't want to wait that long for them to all fall down. But the second reason is when the cells land, it will actually spreads out, and typically whatever the diameter of the cell is, it will double in size. So you have to kind of think in advance, how big would that be once they land. But once you get this right, within 24 to 48 hours, you end up with this monolayer that you see in the middle. It's a perfect monolayer of cells. They're not yet fully polarized, but they are getting there. And then at 48 to 96 hours, you start seeing bulk caliculi. This is where your biologics are being excreted, and that tells you the cells are polarized and ready for your experimentation. All right, I'm gonna speed up a little the presentation here because I realized that I am going to run out of time. So very quickly, so here's an example of what you can do and how you can test it in the real life. So we selected 4 different donors from 3 different vendors. All cells were high quality, they're 90% platable. The COA showed the 5-day culture. They were just great, and we plated them. And guess what? They did not perform equally as they were on the COA. As a matter of fact, the first 2, 1 and 2, were terrible. They didn't plate at all. And 3 and 4 were great, so we could continue with our testing. And because we knew that they work well in our hands with our methods. That doesn't mean that 1 and 2 did not work. It just simply meant that in our methods, in our handling, they just did not perform well. So moving forward, we took those cells and we did some additional testing. We did the synthetic activity for albumin, ammonia metabolism, and did the 3D aggregation for our application. And indeed, number 4 was the best in all cases, and it resulted in a great aggregation and great cells. So all that landed us that we end up choosing this one specific lot. We bought large number of cells, so we put them in a bank, and those were useful in subsequent experiments. Now, one more thing that we did is once we received a large number of those cells in the final shipment, we tested them again, just to make sure that nothing happened during the shipping, nothing happened during storage, and they perform exactly the same way as the first day when we tested them for original testing. That is critical. So again, trust but verify. Once you have those cells, you can generate great results. So because we don't have the time, I'm not going to go into much details about this. This is a really nice study performed by Christopher Dickman, working together with his team, and also Stephanie Campbell and her team at Aspect Biosystems, and they presented that at ASGCT last year and year before that. I really, invite you to go to the ASGCT, look for the poster. Chris actually presented that, and it's recorded, so you can listen to it, how they done it. But long story short on this, they use exactly that method of characterizing hepatocytes to come up with the best quality hepatocytes for the aggregation. Those cells were then printed in 3D and then implanted into mice for treatment of acute liver failure induced by CCL4. And in both examples, you have with the CCL4 acute liver failure, you would expect about 60–70% mortality, which is the red line in the figure. And with the transplantation, he was able to rescue them about 30 to 40%, which is great. And this only because the quality of cells was high, and we ensured that we maintain as little variability as possible. So again, I invite you to listen to his talk and look at this data, and you can email him directly if you have questions about this. But this was a good example of what the good quality cells can generate. And then in the final slide here before my conclusion is, I told you about the spheroid assembly. They typically assemble in 5 days, and they work pretty good. But you can actually speed up the process. One way to do it is mix the cells with other cell types. You can put the hepatocytes with HSCs in small quantity, like the stellate cells or fibroblasts or MSCs, and they can assemble in like almost a day or 2. The reason why is those cells produce ECM, and that ECM helps with this aggregation, and this is really supported. And finally, you can also control the size of your spheroids. You can make very large spheroids for low turnover compounds and use them, for example, for the quick metabolism studies. And you can make them also very small if you're doing this, gene delivery, or you're trying to get the nice images when you don't want a big size. And also you can make them as small as 25 cells, or as large in my experience as 12,000 cells or so. The problem is that with the large spheroids, as they're becoming bigger and bigger, they start developing this necrotic core, and this is the area of the low oxygen tension, and the cells start dying. That results in essentially dead cells that impact the rest of the cell performance and leads to potentially kind of a downgrade of your spheroid. So unless you really care about this, for example, if you're doing tumoroid studies and tumor studies, you don't want to have too much necrotic core, so just be aware of that. So finally, I'm not going to go too much about the conclusion you heard. Hopefully I told you have to trust but verify, test your hepatocytes. The secret sauce to hepatocytes is make sure they have a good viability, make sure that they played well, make sure they polarize by having the bulk caliculi. If you're doing 3D, make sure that they aggregate. And then finally, make sure that they have a basic like synthetic activity, ammonia metabolism. And obviously, if you're looking at SIPs, make sure that the SIPs are expressed well as well. So with that, I just wanted to thank everyone for that they're involved in the work in the past life or work that I did in the past life here. So Thermo Fisher Life Technologies, originally Ambus Medicine, Aspect, and Anna Bios, obviously, for inviting me here today. So with that, I'm open to questions. We'll probably have enough time for maybe a question or two.
Thanks, Raf, for an excellent presentation. So much great information. And just a reminder, this has been recorded, and Gary will be sending out an email to everyone who registered with the recording. So if you missed any details, you can go back and check that out. We do have time for questions, so you can write your questions in the panel. And also just a reminder, you can download some handouts about anabios, hepatocytes, and also cell system LSECs. And I'll get things started. Raf, earlier in the presentation, you talked about pediatric hepatocytes. Obviously, if someone's doing liver tox and they have a specific question about pediatric toxicology, that would be a use for them. Can you describe or think of other situations where pediatric hepatocytes would be useful?
So like I mentioned early, pediatric hepatocytes will contain zone 1 hepatocytes. So if you really care about having all those zonality represented across all your culture, pediatric is your answer. If you're thinking about 3D cultures for longevity, again, those are healthy cells, small cells. They can be much better than doing something that's adult that may be damaged by a disease or damaged by long life or that is ridden with the drugs and alcohol and things like that.
Okay, thank you. And by the way, I've asked several pediatric hepatocyte lots, and we can send you our entire inventory if you send an email to info@anabios.com. So we have several questions from the audience. I'll just dive right in here. Are there any correlations for platability in zone 1 cells? I guess you just answered that.
So typically zone 1 will plate really well because they're very healthy cells.
Right. Yeah. Okay. Is there a preference for 2D or 3D systems for drug or gene delivery?
It really depends on the longevity that you want to maintain in your culture. So if you can assess your cultures with the gene delivery and testing within 5 days, then 2D is probably simpler. But if you're talking about 14, 15 days cultures that you may require, for example, for Hep C studies, then you're kind of limited to really going to 3D or cells that can really last that long in 2D.
Okay. Is trypan blue an accurate method to measure hepatocyte viability?
Yes and no. So there is a way to do the trypan blue. I would suggest that if you really want to focus on trypan blue, do some research on the PubMed and find publications that actually tell you about the concentration of trypan blue that you should use and the time that's required for the cells to uptake trypan blue and spit it out. So the challenge with trypan blue is that the way it works, it's supposed to go to all your cells, but then the live cells are able to pump it out, whereas the dead cells like dead hepatocytes cannot pump it out. Well, the problem is that your hepatocytes are damaged because of isolation. If you put way too much trypan, they're not fast enough to pump it out. So that creates a problem of these false positives.
Gotcha. Another question from the audience, a lot of great questions. Are liquid droplets a bad sign of health for hepatocytes? And can we use them for testing LNP formulated mRNAs translation?
Well, so lipid, I'm assuming the bile canaliculi. So if that's the case, the bile canaliculi just simply involved in telling you that you have excretion to the BSEP and the hepatocyte excreting the byproduct of metabolism. I would say yes, but I don't know. You have to probably look in the PubMed and search the publications for that.
Okay. Is the bile produced by the hepatocyte fluorescent, and does that interfere with any fluorescent assays or flow?
Yes, so a lot of stuff in hepatocytes is autofluorescent, and it is interfering with flow because of autofluorescence. So yes, it is a problem. Typically, what I would suggest, if you can bypass autofluorescence and go to like DAB staining or some other visual staining, I like them better because you bypass this autofluorescence. Otherwise, you have to have just really good controls and be able to subtract autofluorescence from your real signal.
Okay, here's a question from someone in the audience who's having challenges with hepatocyte attachment. Besides collagen type 1, any other recommendations for coating agents?
So fibronectin is good, but keep in mind that you cannot just use pure fibronectin. So you'd want to combine fibronectin with collagen type 1. Also, you can do, you can do simple overlays with gel tricks on your plate. This will provide additional laminin content to the collagen on the bottom of the plate, and you can try with hepatocytes. What I would say is that if you have challenges with attachment, I would check the quality of your hepatocytes or look at your hepatocytes. They should stick, or at least they should stick at the fairly large number.
Okay. In your experience, have you used organ chip systems to culture these 2D or monolayers or 3D spheroids?
Yes. What type of chips?
It doesn't specify. It's just any type of organ chip system. Do you have any that you can recommend?
So I don't have a really recommendation. I think there are different chip manufacturers out there or groups that are working with that, and there are small chips and bigger chips. Typically, the bigger are easier to load. There's more cells, you can get the more relevant data. Remember, the liver is large for a reason. You need to have a large number of cells to really get the good physiological relevance. If you have a very like microchips, like really tiny chips that contain very small number of hepatocytes, you may not get a strong signal or a strong physiological relevance. So you may want to be aware of that. Outside of that, I would suggest go to that paper I suggest you to read about organotypic models and do search on the model on what's out there. I would do exactly the same thing as we do with primary hepatocytes, test them.
Okay. Here's a question for me, actually. It says, does Anabios offer genotyping data on their hepatocyte lots? Yes, we actually did this in partnership with a lead scientist, Alessandro Soto Gutierrez at Pittsburgh. And so we've got some very interesting SNPs in our lots. PNP, for example, is only found in 6% of the population. We have that in over 20% of our lots. So yeah, if you reach out to us, we can send you that information on the genotypes. Then a couple more questions. How many human hepatocyte donors should be used to attain the population variation? It kind of goes back to your topic about variability. And should we use pooled donors for reducing variability?
So I would say this way, it depends how much money you have and time. So in my research, I typically like to have 2 adult and 2 pediatric, male, female. So that gives me kind of enough representation across those 4. And it should be enough for, like, for example, if you're doing, implantation assays or 3D kind of a functional assays. If you're really talking about the population distribution, having pools of 10, 20, 50 might be better for you. But keep in mind the limitations of those is playability may not work. So you may want to focus on just the suspension assays. So you may be limited of how many cells or what type of assays you can use for this kind of for your applications.
Right. Here's another question. When comparing hepatocyte function across plated and spheroids, did you use the same donor lot of hepatocytes?
Yes.
Okay. Have you seen hepatocytes with a high number of liquid droplets, and how do the liquid droplets affect downstream studies? I think you kind of talked about that a little bit before.
Yes, that's bile, yeah.
Yeah.
So typically bile should not affect anything because depending on how you are evaluating your cells, if you're just evaluating the cells where you're looking at the synthetic activity of them or you're doing staining of them, right? I think as long as you know that there will be some autofluorescence and you can bypass that, you should be okay.
Okay.
On the other hand, if you're doing transporter assay, you're actually collecting that bile, and so that might be actually to your advantage.
Okay. Well, let's do one more question. For an experiment using IVT, mRNA, LNP, do you think the LNP would be able to reach the hepatocytes if they're being maintained in the 2D system, or would the 3D system be better?
You know what? It's actually so LNP. So the challenge with LNPs is that number one is you're overlaying your hepatocytes. So you'd want to design your study in such a way that you could provide LNP before you overlay your hepatocytes with some kind of ECM, because otherwise you will trap LNPs in that ECM and you're gonna have reduced delivery. So you have to modify the process. Maybe try to plate hepatocytes first for like the duration of time, then treat them with LNPs for 24 hours and then overlay them, or do the reverse transfection where you put the LNPs on the bottom of the plate before you plate hepatocytes, so then they can uptake it when they're still in a suspension. On 3D is the longevity. So the thing is the same in 3D. If you can start early on, before the cells develop enough in ECM and hold themselves with it, you should be able to get the LNPs inside of the cells fairly efficiently. Now, keep in mind, you have to reduce the size of your spheroids. If you have a large, like 2,000-3,000 cell spheroids, LNPs will not penetrate all the way to the inside. So they're only gonna get on the outside. So you may want to try maybe spheroids of the 50, 100 cells, try to get them transfected. And then if you want them make them bigger, you can actually put them together so they can fuse together and then be larger for your assays downstream. But initially, you do need to start with smaller spheroids and early on.
Excellent. Raf, thank you so much for such a great presentation. It was very well received. There are even questions that we didn't have time to address. I will send those to you so you can get back to the people that asked them. And any of you going to SOT next month in Salt Lake City, please check out the Anabios booth, and I'd like to meet you in person. But thanks again for attending. Raf, thanks for a great presentation. Gary, thanks for putting this together. And we look forward to seeing everyone at SOT or the next Anabios webinar. Thank you very much.
Thank you, Chris. Thank you, everyone, for attending.